Dark chocolate is the rare food that is simultaneously a genuine pleasure and a documented neurological intervention. Not milk chocolate, not chocolate-flavored products, not anything with more sugar than cocoa. Dark chocolate, specifically the type with 70% cocoa content or higher, contains compounds that measurably increase blood flow to the brain within 40 minutes of consumption, that improve cognitive performance on attention and processing speed tasks in controlled trials, and that produce stimulant effects through a mechanism that is meaningfully different from caffeine and does not produce the same crash. The science on dark chocolate and brain function is not wishful thinking by people who want permission to eat dessert. It is a consistent body of research from multiple independent groups using multiple methodologies, finding overlapping results, increased cerebral blood flow, improved cognitive performance, measurable changes in brain activity patterns on imaging that point to a genuine and specific neurological effect from the compounds in cocoa. Understanding why it works, which specific compounds are responsible, what the difference is between those compounds and caffeine, and what cocoa content is required to produce the documented effects changes how you think about dark chocolate from a food into something more interesting. Cocoa, the processed form of cacao beans, contains an unusual concentration of a specific class of plant compounds called flavonols. Flavonols are a subclass of flavonoids, the same broad category of plant polyphenols that includes the anthocyanins in blueberries. But flavonols have a distinct chemical structure and distinct biological activities that differentiate them from other flavonoids. The flavonols in cocoa are primarily epicatechin and catechin, along with their oligomeric forms called procyanidins. These compounds are concentrated in the cocoa solids, the non-fat component of cacao that gives chocolate its characteristic bitter flavor. The bitter taste of high cocoa chocolate is partly a direct sensory response to flavonol concentration. Sweeter, milder chocolate has been processed, diluted with sugar and milk, or made from varieties of cacao that have lower flavonol content. This flavor bioactivity correlation has a practical implication. The cocoa content percentage on a dark chocolate label is an imperfect but directionally reliable indicator of flavonol content. Higher cocoa percentage generally means more flavonols, more bitterness, and more of the documented biological effects. 70% cocoa is a reasonable threshold for meaningful flavonol content. 85% and above is where flavonol concentration becomes high enough to produce robust effects at realistic serving sizes. The flavonol content of cocoa products is also significantly affected by processing. Conventional Dutching, the alkalization process used to reduce bitterness and darken the color of cocoa powder, destroys a substantial fraction of the flavonols. Dutch processed cocoa, the kind used in most commercial baking and hot chocolate products, has dramatically lower flavonol content than non-Dutched natural cocoa. Dark chocolate products are typically not Dutched, but the degree of processing during bean fermentation and roasting also affects flavonol content in ways that are not always reflected in the label. The primary mechanism through which cocoa flavonols affect brain function is the same mechanism identified in the blueberry research: enhancement of nitric oxide bioavailability and the resulting increase in cerebral blood flow. Nitric oxide is a signaling molecule produced by endothelial cells, the cells lining blood vessels. It diffuses into the smooth muscle surrounding the blood vessel and causes it to relax, widening the vessel lumen and increasing blood flow. In the brain, where blood flow is tightly coupled to neural activity through a process called neurovascular coupling, increased nitric oxide availability directly translates to increased delivery of oxygen and glucose to active brain regions. Cocoa flavonols, particularly epicatechin, activate the enzyme endothelial nitric oxide synthase, which produces nitric oxide in vascular endothelial cells. They also reduce the oxidative degradation of existing nitric oxide by neutralizing the reactive oxygen species that would otherwise break it down. The combined effect is substantially increased nitric oxide concentration in the vascular endothelium, increased vessel dilation, and increased blood flow. This mechanism is not specific to the brain. Cocoa flavonols have documented effects on cardiovascular function through the same nitric oxide mechanism, reducing blood pressure, improving endothelial function measured by flow-mediated dilation, and improving blood flow to the heart. The brain effects are the neurological manifestation of a vascular mechanism that operates throughout the circulatory system. The brain is particularly responsive to flavanol-induced nitric oxide effects for two reasons. First, the brain has the highest metabolic rate of any organ in the body. It consumes approximately 20% of the body's total energy, despite representing only 2% of body weight. Its blood flow requirements are correspondingly high and tightly regulated. Second, cognitive performance is directly sensitive to small changes in blood flow to specific brain regions. The prefrontal cortex and hippocampus, where flavonol effects have been most consistently observed, are regions where blood flow is most strongly correlated with performance on the cognitive tasks used in flavonol research. The University of Birmingham study that established the acute cognitive effects of dark chocolate consumption used a carefully controlled methodology that is worth examining in some detail. The study enrolled healthy adults and assessed cognitive performance following consumption of either high flavonol cocoa drinks or low flavonol control drinks in a crossover design, meaning each participant served as their own control. The primary measure of brain function was the blood oxygen level-dependent signal in functional MRI, a measure of regional brain activity based on changes in oxygenated blood flow. Participants who consumed high flavonol cocoa showed significantly increased brain activity in regions associated with attention and cognitive processing, particularly in response to tasks requiring sustained attention and rapid response, compared to the same participants after consuming low flavonol control drinks. The effect was observed within approximately 40 minutes of consumption and was correlated with the flavonol dose. Behavioral performance measures, including reaction time and accuracy on attention tasks, also improved in the high flavonol condition compared to the low flavonol control. These findings are consistent with the mechanism. Increased nitric oxide production following flavonol absorption leads to increased cerebral blood flow within the timeframe of nitric oxide signaling, approximately 30 to 60 minutes. The resulting increased oxygen and glucose delivery to the prefrontal cortex improves the performance of the attentional and executive function processes that depend on prefrontal cortical activity. The effect is acute. It occurs within a single consumption event, not only with long-term supplementation. This is consistent with the finding that flavonols produce rapid changes in plasma nitric oxide and vascular dilation within 1 to 2 hours of consumption. The cognitive effects follow the vascular timeline. The longer-term effects of regular cocoa flavonol consumption on brain function have also been examined. A study published in Scientific Reports followed older adults who consumed high flavonol cocoa drinks daily for 3 months and found significantly improved cognitive scores compared to controls, along with increased brain perfusion measured by MRI. The long-term effects appear to build on the acute effects. Daily flavonol consumption produces cumulative vascular and cerebral blood flow improvements that translate to sustained cognitive benefit. Theobromine is the second bioactive compound in cocoa that affects brain function, and it operates through a mechanism distinctly different from the flavonols and distinctly different from caffeine in ways that most people have not considered. Theobromine is a methylxanthine, the same chemical class as caffeine. Both compounds inhibit phosphodiesterase enzymes and block adenosine receptors, which are the mechanisms through which caffeine produces its alertness-promoting effects. But theobromine and caffeine have different receptor binding profiles, different pharmacokinetics, and different peripheral effects that produce meaningfully different experiential outcomes. Caffeine has a high affinity for adenosine receptors. It binds tightly and blocks the sleepiness signal effectively. It also has significant effects on the sympathetic nervous system, increasing heart rate, blood pressure, and adrenaline release. This adrenergic stimulation is what produces the jitteriness, the anxiety, and the cardiovascular effects that some people experience as side effects. And when caffeine's half-life of approximately 5 to 6 hours leads to its clearance from the system, the adenosine that has accumulated during the period of caffeine blockade floods the now unblocked receptors simultaneously, producing the crash. Theobromine has lower affinity for adenosine receptors than caffeine. It produces milder adenosine blockade. It also has significantly weaker effects on the sympathetic nervous system. It does not substantially increase heart rate or adrenaline at the concentrations present in dark chocolate. And it has a longer half-life than caffeine, approximately 7 to 12 hours compared to 5 to 6 for caffeine. The practical consequence of these pharmacological differences is that theobromine produces a gentler, more sustained alertness effect without the adrenergic stimulation that produces caffeine-related anxiety and cardiovascular effects, and without the abrupt adenosine flooding that produces the caffeine crash. The alertness from theobromine rises more slowly, peaks more gently, and declines more gradually. Dark chocolate contains both caffeine and theobromine, typically in a ratio of approximately 1 part caffeine to 9 parts theobromine. A 70-gram bar of 70% dark chocolate contains approximately 50 to 60 milligrams of caffeine and 400 to 500 milligrams of theobromine. The caffeine provides the initial alertness effect. The theobromine extends and smooths it. Research published in Psychopharmacology compared the cognitive and mood effects of caffeine alone, theobromine alone, and their combination in controlled conditions. Theobromine alone produced improvements in sustained attention and improved mood ratings compared to placebo. The combination of theobromine and caffeine, the profile most closely resembling dark chocolate consumption, produced improved alertness and mood, with less of the jitteriness and cardiovascular stimulation associated with caffeine alone. The comparison between dark chocolate and conventional stimulants deserves explicit attention because it illustrates how the specific compound profile of a food determines its effects more precisely than the category label. Coffee and dark chocolate both contain caffeine, but dark chocolate's caffeine is accompanied by theobromine at a 9:1 ratio, by flavanols that produce independent cerebral blood flow enhancement, by theobromines mood-improving and alertness-extending effects, and by phenylethylamine, a trace amine that affects dopamine signaling and that may contribute to the mood elevation many people experience after chocolate consumption. Energy drinks contain caffeine, typically at concentrations 2 to 3 times higher than dark chocolate, along with sugar, artificial additives, and in some formulations, additional stimulants like guarana or taurine. The high caffeine, high sugar profile produces strong adrenergic stimulation, followed by a pronounced crash as both the caffeine and the blood glucose decline simultaneously. The neurological profile of dark chocolate consumption is a substantially milder, more sustained, and more complex cognitive enhancement than a caffeine-based stimulant. Because the active compounds are more numerous, their mechanisms are additive but different, and their pharmacokinetics produce a more gradual curve. This does not make dark chocolate a substitute for coffee or energy drinks in situations requiring rapid onset of high alertness. It makes it something different, a food that produces moderate, sustained improvements in attention, blood flow, and mood through multiple simultaneous mechanisms, without the adrenergic side effects of high caffeine products and without the glucose crash of sugar-containing energy products. The practical parameters for consuming dark chocolate in a way that produces the documented cognitive effects are specific. Cocoa content of 70% or higher is the appropriate threshold. Below this level, the sugar and milk content dilutes the flavanol concentration to a point where achieving a meaningful dose requires consuming quantities whose caloric and sugar content outweighs the benefit. A serving of 20 to 40 grams of high cocoa dark chocolate, 1 to 2 small squares from a standard bar, provides a dose of flavanols and theobromine in the range that has been shown to produce acute cognitive effects in the research. This is a modest serving that fits within a normal caloric budget. The timing of consumption matters for the acute cognitive effects. If the goal is improved performance on a specific task, a presentation, an exam, a period of concentrated work, consumption 40 to 60 minutes before the task allows the nitric oxide-mediated blood flow effects to peak during the task. This is consistent with the Birmingham study timeline. Daily consumption of a small serving of high flavanol dark chocolate in the range of 10 to 20 grams is supported by the research on cumulative long-term cognitive and cardiovascular effects in attention, processing speed, and cognitive performance in controlled trials. It contains theobromine, a methylxanthine with gentler adenosine receptor affinity and lower sympathetic stimulation than caffeine, that produces sustained, mild alertness and mood improvement without adrenergic side effects or the pronounced crash associated with high caffeine products. It contains the combination of both compounds at a 9:1 theobromine to caffeine ratio that has been shown to produce better cognitive and mood outcomes than caffeine alone. Long-term daily consumption of high flavanol cocoa reduces cardiovascular mortality by 27% in the largest randomized controlled trial of cocoa flavanols conducted to date. 70% cocoa content or higher. 20 to 40 grams per serving. 40 to 60 minutes before cognitively demanding tasks for acute effect. Daily for cumulative benefit. Non-Dutched cocoa powder as an alternative. Dark chocolate is not a health food by virtue of being chocolate. It is a specific food with specific active compounds at specific concentrations that produce specific documented biological effects. The concentration of those compounds is what the cocoa percentage measures. Below 70%, the dilution is too great. Above 70%, the evidence-based biology begins. If this changed how you think about the dark chocolate you have been treating as an indulgence, hit the like button. Share it with someone who reaches for an energy drink when they need to focus. And subscribe for more content like this. See you in the next one.